Bismillahirrahmanirrahim. My name is Humaria Alam, student of M.phil microbiology first semester. Topic of my presentation is high prevalence of clostridium botulinum in vegetarian sausages. Contents are introduction, objective, materials and methods, results, discussion, conclusion, and references. In line with sustainable developmental goals, plant-based foods and meat substituents are becoming a preferred source of protein among increasing number of consumers. A popular alternative for meat products are vegetarian sausages, with a range of products being available on the market. Most products are vacuum packaged chilled foods, but also frozen and canned vegetarian sausages are retailed. A major food safety concern in vacuum packaged chilled foods are psychotropic botulinum neurotoxin producing Clostridia, particularly Clostridium botulinum group 2. These bacteria produces resistant endospores, grow in anaerobic conditions, and produce the highly potent botulinum neurotoxins during growth. Once ingested, botulinum neurotoxins inhibit the release of acetylcholine at the neuromuscular junction and cause potentially lethal plastic paralysis. Botulism. C. botulinum spores exist widely in environment and can contaminate food raw materials. The spores can survive pasteurization and under favorable conditions may germinate and outgrow into toxic cultures. While there are no reported cases of botulism due to vegetarian sausages, the possibility of raw materials contamination with C-botulinum group 1 and 2 spores, mild pasteurization, vacuum packaging, and long shelf life contribute to an apparently high risk of C-botulinum growth and botulinum neurotoxin production related to vacuum packaged chilled vegetarian sausages. Here we show a high overall prevalence of 32% of C-botulinum in 74 vegetarian sausages. This table shows the prevalence of Clostridium botulinum and botulinum neurotoxin genes in vegetarian sausages. In total 74 samples were taken, 66 from the vacuum package and 8 from the frozen. Out of 66 vacuum package samples, 23 were positive. 8 for type A, 7 for type B, 6 for type E, 1 for type B and E, and 1 for type B and F. Out of 8 frozen samples, 1 was positive for the Clostridium botulinum and botulinum neurotoxin genes, and this was for type B. Objective of this study was to check the prevalence of Clostridium botulinum in vegetarian sausages. Methodology. A total of 74 samples of 8 frozen and 66 chilled packaged vegetarian sausages from 7 producers were purchased in Finland and Germany. Before laboratory analysis, the vegetarian sausage samples were stored at temperatures instructed by the manufacturers, either frozen or at refrigeration. The quantity of C. botulinum was determined from non-heat shock samples using the most probable number method, using PCR detection of C. botulinum growth and formula of Thomas for MPN estimation based on the number of PCR positive tubes. A sample size of 20 to 111 gram was inoculated into a set of tubes containing tryptone, peptone, glucose, yeast extract broth, and incubated under anaerobic conditions at 30 degrees Celsius for group 2 or at 37 degrees Celsius for group 1 for 72 to 96 hours, followed by overnight cultures in fresh TPGY under identical conditions. Cells from 1 milliliter aligos were prepared for PCR template uh, as described in Lindstrom et al. 2001. The presence of genes encoding vitamin neurotoxin type A, B, E, and F was determined using multiplex PCR. Attempts to isolate C. botulinum from all PCR positive samples were made on egg yolk agar plates. An amplified fragment length polymorphism method was used to genotype the C. botulinum isolates. The results showed a high prevalence of Clostridium botulinum and botulinum neurotoxin genes in vegetarian sausages. Apart from one frozen positive sample, all the other positive samples were detected among chilled products. Genes for botulinum neurotoxin types A, B, E, and F were detected, with types B 33%, A 33%, and E 25% detected frequently, and type B and F together, and types B and E together once 4%. The highest MPN counts were detected for group 2 type E up to 1200 cells per kg. At the time of purchase, 8 samples had remaining shelf lives of 36 months, and 6 of them were PCR positive for C. botulinum. 8 isolates were recovered from 8 PCR positive samples, 2 types A, 5 types B, and 1 type E isolate. AFLP typing showed all the A and B isolates to be of C botulinum group 1 and the one E isolate to be of group 2. This dendrogram of 8 clostridium botulinum isolates originating from vegetarian sausages and at 25 degrees Celsius. Successful isolation of C-botulinum validated the positive PCR findings, with successful recovery of botulinum neurotoxin genes carrying isolate as the sole criterion of a positive sample. The overall prevalence of C-botulinum in the vegetarian sausages appeared to be 11%. Discussion. Highest C-botulinum counts for C-botulinum group 2 type E, which are cyclotrophic and can grow and produce botulinum neurotoxin at temperature as low as 3 degrees Celsius within 8 weeks were found. The high counts were detected in products with remaining shelf lives at 6 to 10 degrees Celsius for up to 6 months. While vegetables are a common source of group 1 C botulinum types A and B, group 2 type B strains are associated with the pork meat preparations and occasionally to fish and seafood. Type E is mostly associated with fish and seafood. Source for type E could be sea salt, as the sea botulinum type E strains prevail in aquatic ecosystems and their spores have been detected in sea salts. While botulinum neurotoxin A is exclusively produced by C. botulinum group 1 strains, and botulinum neurotoxin type E by group 2, botulinum neurotoxin B and botulinum neurotoxin F toxins can be produced by both C. botulinum group type 1 and 2 strains. The applied PCR from group 1 and 2 strains and group identification was obtained. Only methodology does not distinguish between botulinum type B and botulinum neurotoxin F genes when isolation of C. botulinum was successful. AFLP typing showed all the 5 botulinum B positive isolates to belong to clostridium botulinum group 1. The similar prevalence and counts of botulinum A and botulinum neurotoxin E findings demonstrate that both group 1 and 2 strains are prevalent in these products, and the applied heat treatment did not eliminate any of the two groups. While C. botulinum group 1 strains bear of food safety concern due to high heat resistance of their spores, their growth and toxin production can be controlled by refrigeration. However, group 2 strains, whose spores are of moderate heat resistance, are of concern due to their growth and toxigenic potential at low temperature. The time to toxigenesis varies greatly by multiple factors, including the number of spores, efficiency of the heat treatment applied, storage temperature, and intrinsic factors like water activity and pH. There is no information available on the growth and toxigenic potential of C. botulinum group 2 at 0 to 3 degrees Celsius for extended time periods of 6 to 9 months. To control the safety of vacuum packaged chilled foods with shelf lives over 10 days, it is advised that the product are given a 60 heat treatment, a process that will reduce the risk of Clostridium botulinum group 2 spores to 6 log. It is recommended that additional factors like chilled storage below 3 degrees Celsius, water activity below 0.97 corresponding to water phase initial concentration of 5% or higher, or pH below 5% to be applied to control spore germination and outgrowth. If Mayer fails to control Clostridium botulinum growth and toxin production, botulinum neurotoxin can be destroyed by heating at 85 degrees Celsius for 15 minutes or at 80 degrees Celsius for 20 minutes. 85 degrees Celsius for 5 minutes or at 80 degrees Celsius for 20 minutes. The safety of vegetarian sausage products must rely on multiple hurdles controlling growth and toxin production, and never merely on toxin inactivation during cooking. Conclusion: a high prevalence of Clostridium botulinum in vegetarian sausages suggests that these products could be a potential source of botulism. Mild heat treatments enable survival of Clostridium botulinum group 1 and group 2 spore, and long shelf life may support spore germination, outgrowth, and toxigenesis. Inoculated pack studies and shelf life tests are required to determine the growth and intrinsic potential of C. botulinum in vegetarian sausages and the length of safe shelf lives. These are some of the differences. Thanks for listening.